Hello, my name is Aurora Duncan, and I'm a current master's student at the University of Cape Town. I'm going to be talking a little bit about my thesis today, which is in the variation in wing area and echolocation detection volume of Rhinolophus capensis, or the Cape horseshoe bat, in response to different climates. So climate change is a particularly hot topic right now. But it wasn't until I saw these two images, one from National Geographic and the other from the BBC, that I got really interested in learning about how bats are responding to climate change. Bats are particularly dependent on climatic conditions for foraging, reproduction, and roosting, but humans are also dependent on bats. Compared to the body allow
Hi there. It appears that this talk hasn't been presenting with any sound, so I'm going to begin it from the beginning. Apologies, it's going to eat into the break a little bit.
I'm going to be talking a little bit about my thesis today, which is in the variation in wing area and echolocation detection volume of Rhinolophus capensis, or the Cape horseshoe bat, in response to different climates. So climate change is a particularly hot topic right now. But it wasn't until I saw these two images, one from National Geographic and the other from the BBC, that I got really interested in learning about how bats are responding to climate change. Bats are particularly dependent on climatic conditions for foraging, reproduction, and roosting, but humans are also dependent on bats to provide ecosystem services like seed dispersal, pest control, and pollination. Another important reason to study bats is because they can act as bioindicators. So from my study, I wanted to look at their two most novel traits, wings, which give them the ability to fly, and echolocation, and see how these two traits are adapted to local climatic conditions and how these traits may change due to climate change. Bats, being the only mammal capable of true flight, make their wings quite interesting structures to study. Bat wings serve two purposes, the first and most obvious one being of flight, the second being of thermoregulation. Some studies have shown that the wing capillaries will widen and flood with blood, supposedly allowing heat to radiate from the wings into the surrounding environment. Others have shown that the large, flat surface area of the wings compared to the body allow wings to dissipate excess heat from the body. However, little is known about how the structure of wings are currently adapted to account for thermoregulation within their environments. One relationship that is well known between wings and their environment is that of environmental clutter. Environmental clutter is simply anything in the environment like buildings, plants, trees, or shrubs that bats must account for when they're flying. Relative to their body size, bats with short, broad wings typically forage in more cluttered environments. This allows them to prioritize maneuverability and agility so they can fly between the plants, trees, and shrubs in order to catch their prey. In contrast, a bat with long, thin wings can fly faster but is less maneuverable. However, this is okay in environments with low amounts of environmental clutter because bats must fly faster in order to catch their prey. Given this well-established relationship, does environment type also play a role in determining wing size based on differing thermoregulatory needs? So in other words, will we find bats with larger wings in hotter environments, which would allow them to dissipate heat more effectively, and smaller wings in environments that are colder? Echolocation, a bat's second most novel trait, is also highly variable. Bats employ different echolocation strategies to help them avoid the self-deafening of their sensitive ears from the high-intensity pulses they emit. Low duty cycle pulses are comprised of short, frequency-modulated sweeps that vary in bandwidth and are separated by long periods of silence, while high duty cycle pulses of horseshoe bats are comprised of longer, constant frequency components ending with a short frequency-modulated component and are separated by short periods of silence. The left column shows low duty cycle calls, and the right column shows high duty cycle calls. Now, there's a lot to get into here, but the most important thing to know is that high-duty cycle echolocators use a clutter-resistant echolocation system in which an acoustic phobia is combined with Doppler shift compensation and acoustic glints generated by the fluttering wings of insects to detect prey in dense vegetation. The acoustic phobia is the region in a high-duty cycle bat's auditory cortex with neurons particularly sensitive to a narrow range of frequencies that a bat can hear. Because bats echolocate while in flight, their returning echoes are higher in frequency than the emitted pulse due to the Doppler effect. To ensure that the echo returns at a frequency within the range of the acoustic phobia, bats emit pulses at slightly lower frequencies than those to which the acoustic phobia is tuned, so that the returning echo falls within the range of their acoustic phobia, as a result of the Doppler effect. This phenomenon is called Doppler shift compensation. So here are some differences between low-duty cycle and high-duty cycle echolocation. The most important thing you need to know here is that low-duty cycle echolocators, which comprise the majority of echolocating bats, have a degree of flexibility to adjust the parameters of their calls, including the peak frequency, or the pulse with the strongest energy, the bandwidth, the intensity, and duration of their pulses. So this allows them to live in a wide range of habitats and consume a variety of prey items. Because of their acoustic phobia, high-duty cycle echolocating bats do not have this luxury. They don't have the flexibility in adjusting their echolocation parameters because of the Doppler shift compensation, and this has made them clutter specialists. So they hunt in areas with high amounts of vegetation and shrubs. And because of the acoustic glints that are generated from the flying insects that they can detect while echolocating, there's a high prevalence of flying insects in their diets. High-duty cycle echolocation poses a risk to the foraging efficiency of bats. Firstly, because environmental conditions already play a direct role in the sound propagation. For example, in areas with high humidity, the sound is more readily absorbed. Low-duty cycle echolocators can adjust the parameters of their calls, but high-duty cycle echolocators cannot, due to their acoustic phobia. Increases in ambient temperature due to climate change may also more readily absorb high-frequency calls, potentially decreasing a bat's detection range and affecting a bat's ability to hear the returning echo of its prey. So in other words, high temperatures already are absorbing the sounds more readily. But because high-duty cycle bats cannot adjust the frequency or intensity or bandwidth of their calls, they cannot respond to these environmental changes. So some studies have shown this phenomenon to be true. One in particular by Luo et al. In 2013 showed that a bat's prey detection volume would decrease when temperatures increased by 2 to 4 degrees Celsius. I wanted to ground truth this study by looking at the Cape horseshoe bat across its range. So something else that is particularly important to note is that bat wings and echolocation form an adaptive complex, meaning any selection on wing area or echolocation detection volume is likely to influence changes in the other, with consequences for the foraging efficiency of bats. My question: will climate change have impact on the foraging efficiency of the Cape horseshoe bat? This is the Cape horseshoe bat. It is a high-duty cycle echolocator with different peak frequencies across its range. It occupies a variety of ecosystems and biomes across South Africa, which you will see in the next slide. And something particularly important to note is that what I looked at was the potential impact of climate change on the foraging efficiency of this bat, gauged by this bat's adaptive responses to different climatic conditions over its geographic range. This is a Cape horseshoe bat's geographic range. You can see it goes all the way up to the border of Namibia in the Richtersveld, which is an extremely hot and arid environment, down through the Western Cape, and back up through the Eastern Cape, which is known for its dense forests and shrublands. So these are the different methods that I use to answer my question. So first, we set up mist nets and harp traps, which were used to catch bats after they returned from foraging. Once we captured the bats, we placed them in cotton bags, and the next day we took different biological parameters, such as sex and forearm length, and importantly, we took digital images of their wings outstretched. Next, we used a microphone array system, which was used to record the bats upon exiting their roost to forage. And we continually monitored environmental conditions using a weather station. So from the wing images that we took, I created a linear mixed effects model to account for the possible effects of body mass, environmental clutter, latitude, environmental temperature, and sex. This is what one of the images looked like. My results showed that body mass was the only significant predictor for wing area, which isn't too surprising given that larger bats need larger wings in order to generate lift and fly. So as you can see here, wing area increases with body mass. From these results, I can conclude that wings do not likely function as a thermoregulatory appendage. Bats likely use other methods in order to dissipate excess heat, such as panting or licking their bodies. But what was surprising was that there was no relationship with NDVI or clutter, given that there is a well-documented relationship between wing shape and clutter, but no relationship between wing area and clutter. Finally, changes in ambient temperature are still likely to affect wing area due to allometric changes in body size, as described by something like James's rule, but this should be explored in future studies. From the microphone array system, I was able to use MATLAB to generate these flight paths, which showed exactly where the bats were flying in a 3D space. Then I extracted values for source level and frequency, which I then put into this equation to generate the values for prey detection volumes. This is from the Luo et al. Paper. Then I created a linear mixed effects models, which took into account the possible effects of environmental clutter, latitude, and environmental temperature on echolocation detection volume. And from this, I found that average winter minimum temperature, latitude, and NDVI, or clutter, all had effects on the echolocation detection volume of the Cape horseshoe bat. As you can see here, echolocation detection volume decreases with increasing average winter minimum temperature. Echolocation detection volume increases with increasing latitude, and echolocation detection volume decreases with increasing NDVI, or clutter. From these results, I can conclude that there's evidence to show that changes in ambient temperature will directly affect the prey detection volume of bats. This shows support for the Luo et al. Paper that I was hoping to ground truth. Also, changes in temperature will likely lead to changes in vegetation type as well. And because NDVI also affects prey detection volume, this will only add to that effect. So, it is apparent that the wing area and echolocation detection volumes of the Cape horseshoe bat are adapted to local climatic conditions. Changes to environmental conditions pose a risk to these animals that are reliant on acoustic signals. And while both traits are likely to be affected by changes in climate, the adaptive complex that they form make changes in one likely to affect the changes in the other. The rapidity of climate change makes adaptation to these new conditions unlikely. Going forward, it is important to be aware of species that are less adaptable than others and act to conserve areas where these species are located. Furthermore, it is important to understand the complex relationship that exists between traits, in this case between wing size and shape and echolocation type. Thank you for listening to my presentation. I hope that you all have a great virtual conservation symposium.